Hey everybody, it's Mrs. Mancuso with some more AP Bio review. This time I want to talk about electrophoresis. Electrophoresis, and actually want to talk about genetic engineering. So, genetic engineering is when you go through and you change your DNA. And you do that by adding something to it. So if you want to add a gene, you can genetically engineer it. I always go back to the example of the Honeycrisp apple because, I mean, what's a more perfect apple? They took the big size, the perfect redness. It looks amazing. You walk into Wegmans and you see it and you have to buy a Honeycrisp apple. And then you taste it and it is crunchy and it's sweet and it's tart and it's just awesome. So they took all the genes and they put them together. So how do you do that? How do you genetically engineer something? First of all, you have to have restriction enzymes. Restriction enzymes cut DNA, and they cut it at specific areas called palindromes. And a palindrome is a section of DNA where it has a repeating pattern where it's the same if you go in the forward direction or the reverse direction. So we have a lot of different restriction enzymes. And what it does is here's my DNA and here's all of my base pairs. So this would be like an A with a T and a C with a G. And what it does is it goes in, I'm going to use a different color. It goes in and it cuts across and it cuts down. And so what you end up getting if you cut it like that is a piece like this. That's called a sticky end. It's exposed. And this is still together. And then you have another piece here, sticky end, that's exposed. I can now give it another piece. So here's another piece that I also cut. So it's got an openness. And it's got a together part, and it's got together and an open. And what will happen is it will come together. This will take up this piece. This will take on this piece. And these can actually join too. I just don't have room on my paper. But this can all reconnect. So I end up having this long piece of DNA that's got some blue section and red, like that. And this has my whole blue section and red. And this is my new gene. I've inserted a gene. You need ligase. Ligase is the enzyme that'll seal it back together, but I now have recombinant DNA. And you might remember in class, I did this with two necklaces. I opened up one necklace, I opened up another necklace, and then their ends were exposed, so those were my sticky ends, and then I put it back together. And when I put it back together, I inserted the gene. Now when transcription happens, this is gonna open up. RNA polymerase is gonna read this gene and make RNA, and now I can express it. And so if I am expressing a new gene, I am genetically engineered. I have changed. All right, electrophoresis. Electrophoresis is a test that we do where we set up a gel. So here's my gel, and my gel has to have an electric current going through it. I know that DNA is negative, so this is my negative end. The place where I put the DNA is negative, and this is my positive end. And once my gel is ready, I'm actually going to turn on electricity so that the fragments will move. So my first rule is I have to treat it with restriction enzymes first, because that's going to cut the DNA into pieces at recognition sequences. I always run a standard. If I want to know the sizes of my other gel, my other fragments in the gel, I have to run a standard. A standard, I know how many fragments I'm going to get, and I know how far they're going to travel, because I don't know how far they're going to travel, I know their size. So let's say my standard is a piece of DNA like this, and I cut it twice. That gives me 1, 2, 3 pieces. 2 is the smallest piece of all. That means it will travel the furthest, so this would be piece 2. Piece 1 is a little bit larger, so that will travel right there. And piece 3 is really big, so it's going to be up here. It didn't travel very far. I know the size of 2, 1, and 3. Let's say I know that this is 525 base pairs, and I know this one is 823 base pairs, and I know this one is 17,500 base pairs. What I can do is I can now use logarithmic graph paper. The difference about that is it goes 100, 1,000, 10,000, 100,000. And this would be millimeters traveled. So I go ahead and I measure. What's the distance that this one traveled? So it didn't travel very far, and it was 17,000, so I plot the point. So let's say this traveled 2 millimeters. The distance here is 2 millimeters. And then the distance from the well to number 1, let's say that that traveled 16 millimeters. So I go over here to 16 millimeters, and I know it was 823 base pairs, so I mark that point. And then let's say this one traveled this far. This was 18 millimeters. So here's my 18 millimeters, and I know that was 525 base pairs, so I mark it right here. Then I make a best fit line. All right, now I can run whatever I want up here. Here's my sample 1, and here's my sample 2. And I don't know much about these samples. I am running a gel to really learn about these samples. So what I get from 1 is a piece here and here. So what that tells me is that my original piece of DNA was only cut once. If you get 2 pieces, your restriction enzyme only had 1 recognition sequence, and it cut it into 2 pieces. The 2 pieces are somewhat close in size, and this is their distance that they traveled. So when I turned on electricity, fragments move based on size. These 2 have many more base pairs than 1 and 2 because they didn't travel as far. They have fewer base pairs than number 3 because they traveled further. I would measure the distance. So let's say this one traveled 4 millimeters and this one traveled 6 millimeters. If I want to know how many base pairs are in those fragments, I go down here to my graph, I find the 4, I go up to the line, and I go over, and obviously I don't have good graph paper here, but I can now read this and figure out how many base pairs are in this piece. I can go here to the 6, go up and over, and figure out how many base pairs are in that piece. Now, you do gels for multiple reasons. There are 4 basic tests that are done using gel electrophoresis. The first is crime scene analysis. So if you're doing a crime scene analysis, I'm expecting the pattern to be the same. If this is my suspect, this person's my guilty because they have the exact same cuts. So that means they have the same sequences and the same recognition spots. And I got the same banding pattern. If I'm doing paternity, I'm going to run mom, I'm going to run dad, and I'm going to run the child. The child's genes have to match up with dad and mom. Some of them are coming from dad, some of them are coming from mom. If the 2 together equal the child, then you have the right combination of parents. All right, 2 more things that they're used for. Evolutionary relationships. So I love to show a picture of an okapi. You can Google it if you forget what it looks like, but it's a really funky looking organism. Looks sort of like a zebra and a horse. And so you try and figure out what's it related to, and you can't really tell by just doing comparative anatomy and looking at its overall external look. You compare its DNA. And so you'll look at specific chromosomes and specific genes and look at their sequencing, and you can figure out who it's most related to. And it's related to a giraffe and a horse, I think it was. My gosh, I'm forgetting. But you would do that DNA test to see. And then, so that was a 3rd thing it's used for. A 4th thing it's used for is to detect genetic conditions. So if I wanted to see, let's say my family had a history of breast cancer, they actually know the sequence of the BRCA gene, and they know where it cuts and how many fragments you get. So if they know the BRCA gene and they know it looks like this, and they run me and I come out like this, they know I have that gene. But instead, if I came out like this, I don't have that same pattern, so I don't have that gene. All right, let's go into a little bit more genetic stuff. Let's talk about cloning. So cloning is what happens. We always think of Dolly, the sheep. So when you clone something, you want to make an identical copy. And so what you want to do is you want to take an egg and you want to take out the nucleus. The nucleus is haploid. I want it out of there. And then I have my donor, and I want to take a body cell. And my body cell is diploid. So I want to take that diploid nucleus out and put it in the egg. So here's my egg from one parent, and here's my donor nucleus from another parent. And I now want to put this egg back in the egg organism that it came from. So I implant it back in, and it grows, and it becomes a baby. Now, this baby is going to look just like and be identical to this donor because it received all the DNA from the donor. All right, so that's what cloning is. A lot of ethical implications, like what should we clone, what should we not clone. If there was ways of cloning different organisms so that you would have the right MHC markers and you wouldn't reject something, that would be pretty awesome. So they're working on figuring out what we can and can't do with that. All right, here's another thing, Rflps. Rflps stands for restriction, fragment, length, polymorphisms. So these are how you can tell differences between members of the same species. So let's say I'm looking at a bunch of different humans. All of our DNA is so similar. So I gotta look for introns. I gotta look for areas where we are different and run those on a gel. If I don't look for Rflps, every gel I run is gonna come out the same because we both have hemoglobin, we both have 2 eyes, we both have eyelashes, we both have the ability to make different pigments. So you gotta look for something different. So they use Rflps when they're trying to find differences between humans. All right, thanks for joining me for review.